All right, in our last screencast for Chapter 8, we're going to look more closely at regulation of enzyme activity. Okay, it is super important to regulate enzyme activity. Okay, this helps control your metabolism. And again, basically, chemical chaos would result if a cell's metabolic pathways were not very tightly regulated. And cells regulate their pathways by switching on or off the genes that encode enzymes, or by regulating the activity of the enzymes. So we're going to look a little bit, we talked briefly in the last screencast about how to regulate activity of enzymes with cofactors and environmental conditions and inhibitors. So we're going to look a little bit more specifically at that in this screencast. Okay. So we're going to talk mostly about allosteric regulation. So allosteric regulation is interesting that it can either inhibit or stimulate the activity of an enzyme. So it can either turn it down or turn it on and up. So allosteric regulation occurs when a regulatory molecule binds to a protein at one site and affects the protein's function at another site. Okay, so it is a non-competitive interaction. Okay, it doesn't interfere, or it doesn't bind at the active site. Okay, so most allosteric regulated enzymes are made from polypeptide subunits, which means they have quaternary structure. Okay, so they are large, complex enzymes. Each enzyme that's allosterically regulated has both active and inactive forms. Okay, if the inactive form is bound by an activator, that stabilizes the active form of the enzyme and turns on. So activators turn it on. If an inhibitor binds, this stabilizes the inactive form and turns it off. Okay, so we'll have both allosteric activators and allosteric inhibitors. Okay, so here's how they work. So if we have, here's my active form, and here's my inactive form down here. And we have oscillation here, so it kind of just goes back and forth between these two forms. If we add an activator molecule right here, it will stabilize the active form and turn your enzyme on. Okay. If we add an inhibitor, okay, it stabilizes the inactive form and turns it off. Okay, so different types of allosteric regulation. Cooperative is one form of allosteric regulation that can actually not just turn it on, but turn it up. Okay, it can amplify enzyme activity. And this involves binding of the substrate. So basically what happens in cooperativity is one substrate molecule binds and primes the enzyme to act on additional substrate molecules more readily. Okay, so it binds, one molecule binds in one active site, and it increases the efficiency of the enzyme to bind other molecules. Okay, cooperativity is allosteric because the binding by a substrate to one active site affects the catalysis in a different active site. So away from the active site, different location. Okay, here's how cooperativity works. So basically the substrate can bind, and this stabilizes the inactive form and allows it to be easier for more substrate molecules to bind in other active sites. Allosteric regulators, whether they're activators or inhibitors, are very attractive to drug candidates for enzyme regulation because they are so specific, because you can target a specific enzyme to turn on or off. Another type of allosteric regulation is feedback inhibition. Feedback inhibition is where the end product of a metabolic pathway feeds back into the pathway at an earlier time and can shut down the pathway by regulating an allosteric enzyme. Okay, and feedback inhibition basically prevents a cell from wasting time and money by synthesizing more product than is needed. So let's look at a pathway. So basically, this is the synthesis of isoleucine by this metabolic pathway. So you start with the initial substrate is threonine. Okay, and again, this pathway would be endergonic because I'm making something, and so it's costing energy. So the pathway has a number of steps and a number of enzymes. So there's 1, 2, 3, 4, 5 steps and 5 enzymes that catalyze each of those steps. Okay. So what happens is you go through here, the first enzyme takes threonine, turns it into some intermediate, second enzyme turns it into another intermediate, third enzyme, blah, 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 et cetera, and so on, until we have our end product, which is isoleucine. Now, the cell only needs a certain amount of isoleucine at any one given time. And because this pathway takes energy, we don't want to just keep making isoleucine day in and day out. Once we have enough, we want to shut the pathway down to conserve energy. So what happens is when isoleucine builds up, okay, it feeds back into the pathway at enzyme 1. Isoleucine is an allosteric inhibitor. It binds to the allosteric site away from the active site, and it causes the active site to change shape, so it no longer works. Okay, and this will shut down the pathway, so the enzyme can no longer function in the first step. Okay. If isoleucine is all used up by the cell, it will start to take the isoleucine from the allosteric sites, free them up. That allows the enzyme to convert back into its normal form and proceed to catalyze the first reaction here and make more isoleucine. Okay, so feedback inhibition takes the end product, feeds it back into the pathway to inhibit an allosteric enzyme. And this usually occurs at an allosteric enzyme towards the beginning of the pathway. Okay, another way that enzyme activity can be regulated is basically by location. Okay, so structures within the cell help to bring order to metabolic pathways. Okay, enzymes can act as structural components of membranes, and basically the compartmentalization of the enzymes allows their activity to be controlled. They can only catalyze reactions when they're in the right spot and when their substrates are in the right spot, and so that way we can regulate the activity. Okay, in eukaryotic cells, enzymes reside in specific organelles. So, for example, the enzymes for cellular respiration are located in the mitochondria. So if we look at our mitochondria, okay, there are certain enzymes that are in the matrix and different enzymes that are embedded in the membrane. And that helps us to compartmentalize those two specific pathways and make them easier to control. Okay, and that's it for chapter 8. Hopefully you understand a little bit more about energy transfers, and how enzymes help catalyze metabolic reactions.